So let's begin. So for T cell activation, T and B cell activation, okay? So we said we have the APCs, and I said these are antigen presenting cells, and they are the bridge between the innate. So if you have the innate immune system on this side, and you have the adaptive here, okay? So the APC are the bridge between the two. Okay, so they carry information because the innate are the ones at the battlefield fighting the infection that first comes in and trying to understand what's going on. They are in the trenches, all right? But the APC now take the information to the lymph node. Okay. So if I was to give you a broad overview, let me show you an image here. So this is a good image to show you. Okay. So look at it. So you have, okay, you have your dendritic cell up here. Your dendritic cell. So it's coming from the battlefield, right? The dendritic cells are part of the innate system, immune system. Okay. They don't participate in killing the pathogen. All right. What they do is they gather intel. They gather intel on the pathogen and take it to the secondary lymphoid organ. This is a lymph node. All right. So look at it. Look at the antigen on the surface right here. Okay. It has picked up the antigen on its surface and it's taking it to the battle station. The lymph nodes are like the battle station. Okay. And going to report the information of what's going on, of what it has found to your CD4 as well as your CD8 cells. All right. So antigen record, that's why it's antigen recognition going on in the lymphoid organs. All right. So once it stimulates, it finds a CD4 that can understand the information that it's carrying. All right. It's just like you imagine a military base. You have a soldier who's listening to the radio, trying to intercept enemy communication. He gets an encrypted message. He doesn't understand what it is. That soldier takes it and runs to the headquarters and says, "Oh, I found an encrypted message, intercepted an encrypted message, and I need someone to read it and understand it." And then you find an officer, a superior officer who can do it, the commanding officer who understands that message. And then that commanding officer, you can imagine him like the CD4 positive T cell. That commanding officer will now become activated, all right? Become activated and then go out and migrate or stimulate other white blood cells, all right? Look at it here. So the effector CD4 cell will now recruit other cells, all right, like macrophage, for example, here, to kill the pathogen, all right? Or the CD8 positive T cell to go there and go and destroy the pathogen. So this is a broad overview of the whole process of the B and T cell activation. So you have an antigen presenting cell, right? Will bring the antigen to the lymph node, meet with the CD4, because CD4 is the most important cell to coordinate the immune response, not the CD8. The CD4 is the most important one. And then when the CD4 is activated, it will form effector cells as well. Look at it, it will form effector cells and memory cells. All right. Effector cells and memory cells. Now, what's different? The effector cell is the one that leaves, okay, and goes and mobilizes other white blood cells to do all these things to kill the pathogen and organize and coordinate inflammation and all of that to destroy the pathogen. But the memory cell remains behind in a and it has received that information. It's in a heightened state of awareness about the pathogen, and the next time that pathogen comes around, after the pathogen has been gotten rid of, the next time it shows up, the memory B cells will be readily activated. Both this happens for both B cells as well. Okay, and this when we get to vaccination, this is what vaccination is trying to accomplish. All right, it's trying to accomplish the production of, besides the antibodies that are to induce in the system to combat any of the infection that are caused. But the main thing thing about vaccinations, you're trying to induce memory B and T cells. You're trying to educate the body, like to be, what the old saying, to be forewarned is to be forearmed. All right. So sort of forewarn the body about a potential pathogen, and then the body becomes ready for it. And how does it get ready? By creating memory and B cells. Okay. So let's go back to first aid, to come back to. So now that we've seen that overview, all right. So the antigen presenting cells are your B cells, dendritic, Langerhans cells, macrophages, all right. So those are the antigen presenting cells. So they take the information wherever they get it and bring it to the T cell, particularly the CD4 T cell, all right. So the CD4 T cell, most important cell to coordinate the immune response. All right. So now look at this information here. They say two signals, very important. Two signals required for T cell activation. All right. Two signals for T cell activation, as well as B cell activation and class switching. So we'll get to these concepts. All right. So for T cell activation, all right. So look at what's going on here. So from what I said earlier, from that diagram said, the antigen presenting cell has come in the lymphoid organ and has met the T cell. All right. So this is your antigen presenting cell. This is your T cell. All right. And when we say naive, when you hear the term naive cell is a cell that is, to use the complex, I'll use a complex term and explain it. It's a cell that is a T cell that is immunologically inexperienced. Okay. Immunologically inexperienced, meaning that it has never encountered any antigen. Okay. Never encountered any antigen. So that's what a naive cell. It's a mature cell. It's mature, meaning it has gone through the thymus, passed the test, the screening test in the thymus, and it's in the secondary lymphoid organ, that's the lymph node, and but it has not encountered any antigen. Okay. Okay. So that's a naive T cell, right? Now, so that's the cell that is available to respond to the antigen. All right. So it is not a T cell that has already been activated. Maybe imagine you had maybe a throat infection from strep, whatever kind of infection you want to think of, and then immune response has been activated against it. But the antigens were taken from that infection, presented to a T cell. That T cell became activated. It's not that activated that was responded to a different one infection that is now available to respond to a new or different infection. No, it's a freshly minted T cell, right? A new T cell that has a new graduate from the Thymus Academy, okay, that has just reported to the battle station in lymph node. That's the T cell that's available to respond to the antigen. And that's why when we're talking of immunosenescence, when you don't have these cells, okay, to respond to this antigen presenting cells. So it's like the antigen presenting cells carrying this, just like I talked about the soldier, the recruit who intercepted a intel encrypted message of the enemy and is running around in the battle station or in the headquarters looking for someone to interpret, and he cannot find any commanding officer to read it. Same thing here. If you don't have any naive T cell to look at um, this antigen that the antigen presenting cell has, then this body will not respond to the antigen. All right. And that's where the problem in immunosensing comes, where you don't have the naive cell. So let me not digress a bit. Let me not digress too much. So let's coming back to this T cell activation now. All right. So the naive T cell is there, right, and binds to the 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 antigen on the antigen presenting cell, via the MHC. Since it's antigen for CD4, you're talking of MHC2, right? So, so the T cell receptor, as you can see here, so the T cell receptor of the naive T cell, right, binds to the MHC2 on the antigen presenting cell. So this MHC2 is on the surface of the antigen presenting cell. You have the T cell receptor. Okay, your CD4 CD8 is a co-receptor. All right, but it's needed for that interaction to stabilize the reaction between the CD4 between the T cell receptor and the MHC2. Okay, so that reaction is necessary. It's very important. And then, as we'll see when we get, okay, let me not talk about that. So you have the antigen binding to the T cell receptor, right? This is the first signal, the number one signal. The number two signal, co-stimulatory signal, is the binding between CD8 on the T cell, all right? CD28 rather on the T cell and CD80 or 86 on the APC or B7. Now this signal, they say co-stimulatory, but it's extremely important. All right. If you have anything that disrupts this, right? When we're done here, I'll show you another page in a different part of the book, First Aid, that discusses the things that the clinical scenarios that can disrupt this or drugs that are given to disrupt this interaction, which with the clinical implications of those of what of the anything that disrupts this interaction. All right. So if anything disrupts this interaction, this T cell will not be activated. Okay, this T cell will not be activated. In fact, if it binds to the antigen here, right? If the antigen is between MHC2 and the T cell receptor, if you have that number one interaction, but the number two co-stimulatory doesn't happen, this T cell will go into energy. It will go into energy. All right, it will go into energy. So let me. And once I'm done with this, I'll explain what energy it means shortly. But if you don't have the second co-stimulatory signal, but only the first one between MHC2 and the T cell receptor, energy will happen, right? Meaning that the T cell, the short meaning of energy is the T cell will fail to respond to the antigen. Okay, the T cell will fail to respond to the antigen. So that means no. Let me clean this stuff up. So no response to the antigen or presented antigen. Okay. So if you don't have the second co-stimulatory signal, which is very important, then the T cell will not respond to the antigen that is being presented to it. So that's why this co-stimulatory signal, that the signal one and signal two, and as you can see here, it said two signals are required. So if you have only one, but you don't have the second one, the T cell fails to respond. So it will be seeing it, the antigen, it's seeing what the information is being presented, but it doesn't do anything about it, right? And so that process is called energy. So that means when you hear energy, just understand that this is the basics, and the co-stimulatory signal failed to happen. All right. So the T cell was presented the antigen, but the second signal did not happen, and so it failed to respond to it. So based on what we said the APC ingests the antigen here and processes it, will process the antigen and migrate to the lymph node, as we've seen, and then present it to the T cell, all right? And then, so that's the first signal, all right? So the exogenous antigen represented on MHC2, as we've seen here, right? On MHC2 are recognized by T cell receptor on CD4. So this is your T cell receptor, this is your MHC2. Okay, let's clean some of this, all right? Let's clean some of that, okay? So, and then, however, endogenous antigens, so endogenous as the cell produces the antigen. right? And will process the antigen and present it on MHC1 for CD8 positive T cells. Okay. All right. So now the when the first interaction has happened, the next one, co-stimulatory or signal 2, must happen for the T cell to be activated. All right. Like I said, it must happen. So the second signal, which is a co-stimulatory signal, occurs via the interaction between B7, CD8 CD86, okay, and CD28 on a naive T cell. All right. So that is the second signal. So once those two happens at the same time, all right, they must happen together at the same time, then your T cell becomes activated. All right. So and then the activated T cell will produce cytokines. Okay. So now, like I said, if we go back to what we talked about before we move forward, right? So before I move on, let me just say I want you to know that this MHC that the cell is presenting, if it's CD8, you're not talking of antigen presenting cells, right? You're talking of every other nucleated cell. We said CD8 kills viral infected cells, tumor cells, all right? So that will be every other nucleated cell besides APC. APC2 can present to CD8. So, but if it's CD8, you're talking of MHC1, not MHC2. If it's CD4, so CD8, like they've said here, is MHC1. CD4 is MHC2. So I want you to note that. All right. So note that difference. So if it's MHC1, you have if it's MHC1, you have CD8 here. If it's MHC2, you have CD4 here. Okay. So it's not so that's the difference I want you to pay attention to. All right. So depending on the type of T cell responding to the antigen. Now we said that I wanted to say something. So the activated T cell produce cytokines, and the cytokine that it produces depends on the stimulus that it received. All right. So yesterday we talked about the stimulus here that the CD4, the naive CD4 helper cell will receive. Okay. So if it was stimulated by interferon gamma interleukin 12, it will become a Th1. If it's interleukin 2, interleukin 4, it becomes a Th2. If it's transforming growth factor beta interleukin 1, it becomes a Th17. Transforming growth factor beta interleukin 2 becomes a T17. All right. So the different scenarios will determine which type of activated, because all these cells are activated T cells, right? Well, except for uh, this last one, but all of them are activated. They are activated T cells. All the cells are activated T cells, and they produce unique cytokines, right? So all of them are activated T cells. Now, when the cell is activated, now let's look at B cell. Sorry, let's look at B cell activation. All right. So let's narrow down to B cell activation and class switching. Okay, so we have the T cell activation that happens as above. So that T cell comes down here. You have the B cell will ingest its antigen. All right, so let me show you an image of that B cell surface receptor. All right, let's see. So the B cell surface receptor. So this is the surface of the B cell. All right, so you can see this is immunoglobulin M on the B cell surface and binds to the antigen. All right, so this is the structure of of the antigen receptor that's present on the B cell. All right, so that's the immunoglobulin M that will binds to the antigen that you're seeing up here. All right, and once it binds it, the B cell will internalize that antigen. All right, process it. So the B cell receptor mediated endocytosis, that is to take up or swallow the antigen. All right, and then process the antigen and present it on the MHC2. Because remember, we said B cell is also an antigen presenting cell. So to take up the antigen, present it to the T cell, all right? Then the T cell, that's the CD4 positive cell, all right? And the CD4 then, while it's presenting that to the CD4 positive T cell, the second interaction that is going on is between CD40 on the B cell and the CD40 ligand. So this interaction is very, very important, okay? Because you could get questions that will ask you what will happen if you have just the first interaction. This is number one, right? And you don't have this number two interaction. So mind you, we said this CD40 and CD4, it's not just the B cell alone. It happens to the macrophage, happens to the dendritic cell as well. All right, this CD40 and CD40 ligand, it's not just the B cell, it happens for both the macrophage and the dendritic cell. All right, it's also very important. So all those interactions are also happening not just with they're also happening between the macrophage or the dendritic cell and the CD4 positive T cell. All right. So, like I said, the exogenous antigen is processed and presented on the MHC2, which is recognized by the T cell receptor on the TH cell. Okay, so the CD40, as I said here on the B cell, binds to CD40 ligand on the T cell. So, what's the result of this interaction? All right. Once this binding happens, the T cell will secrete certain cytokines. All right. Will secrete cytokines, which you can see here. And then these cytokines will stimulate the B cell and activate it. All right. Which will undergo, the B cell will undergo class switching and affinity maturation. So let's talk about this now. What do we mean by class switching and affinity maturation? Okay. So when we talk of, mind you, this is all happening in the lymph node, right? This is all happening. I said B cells are located in the lymphoid follicles. Okay, T cells in the paracortex. Okay, so when these T cells are stimulated, they'll go to where the B cells are, and you interact with them and stimulate those B cells to undergo class switching. So we say class switching when the B cell initially encounters an antigen that you see here. All right, they'll first produce on the first interaction. Okay, the B cells will produce IgM, which they say is the primary response, immune response. Okay, that's the first interaction with the antigen. Now, when it processes that antigen and then has presented to the T cell, like you see here, all right, this interaction here, and then the CD40 on CD40 ligand on T cell binds to CD40 on the B cell, then the B cell will now be activated to now begin producing other antibodies, IgG, IgA, IgE. All right. So we said earlier that there are 5 classes of antibodies. Okay, we know that D is not secreted, so we have the other one. So the first one, this M, is secreted at the primary immune response. Okay, so when the B cell has met the CD4 cell and has been stimulated by CD4, and then now starts producing, switches from IgM and starts producing IgG, IgA, or IgE. That's what we call class switching. It means that the B cell switched from one class of antibodies to another class. So in this case, switch from IgM to either IgG, IgA, or IgE. That's what class switching means. All right? That's what class switching means. Now, for the other term, affinity maturation. Right? Affinity maturation means that the binding strength, okay, between the antibody and the antigen is increased. Right? So the strength of binding between the antigen and the antibody is increased. And what's responsible for this? This is an outcome. Okay, what's the process that underlies the increased binding between the antigen and the antibody? We call that somatic hypermutation. Sorry, I'm using big words. Okay, but we'll still, I'm just explaining because we'll still, when we get to antibodies and their structure, they will still talk about this. So I'll just, I'm trying to explain them now. So when you see them later, it's not too confusing. All right, so somatic hypermutation just means that if you have the B cell, when the B cell is stimulated following this interaction that's happened here, right? The B cell, for it to, for that, for it to produce antibodies that have a higher affinity for the antigen, it literally has to go and start rearranging the genes that produce the 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 antibody, right? So if you imagine the genes segments that make up your antibody, right? You have this. I'll use different colors. Okay, you have this. All right, let's use another color. You have this. Okay, you have this. Okay, and let's say this one produces. This is the antibody. All right. So let's say initially you had blue up here, you had green down here, you had yellow, or sorry, my colors are mixed up. Okay, you had orange down here, and it bound to a unique set of antigen. All right, this is what the kind of antibody it was producing the before it was activated. Now, after this activation happens with the interaction between B and T cell, it will produce an antibody that has arranged its gene segments that you see possibly it will rearrange it, okay, the gene segments to look different, okay, something like that. It will change the antibody structure, the parts of the antibody that binds to the antigen. It will change it. It will rearrange the segments of that part of the antibody that binds to the antigen. Okay, and so this part will have a higher affinity compared to this. So this antibody will bind more strongly compared to this antibody because it rearranged the genes that it used to make the. So that's what we call the somatic hypermutation. It's literally reconfiguring the genes that it uses to make the antibodies so that it will produce antibodies that have a higher affinity. So this class switching and somatic hypermutation, where is it happening? It's happening in the lymphoid follicles. Very important for you to know. So this class switching, okay, and somatic hypermutation, they are both happening in the lymphoid follicles. And then when you look at the lymph node, so there are several outcomes that happen when the B cell is activated. Number one, the B cells will start to proliferate and increase in number. So they'll proliferate and increase in number. So that makes the germinal center become bigger. All right. So because the germinal center is bigger, all right, it increases in size. It makes the lymph node to get bigger. So you have to lead to enlarged lymph nodes or lymphadenopathy. All right. So you can imagine someone that has a cut in his wound, a wound in the leg, then the antigen presenting cell takes it the to the B cell, or the antigen floats all the way to the lymph node, draining lymph node, and B cell picks it up, produces some immunoglobulin M first against it, and then takes that antibody, presents it to the T cell. The T cell now stimulates the B cell. The B cell starts to proliferate. So the germinal centers go from primary follicles, I mean, the lymphoid follicles go from primary follicles to secondary follicles. The secondary follicles have germinal center. That's where the B cells are literally expanding. They're increasing in number. Okay. So that's the second thing that happens. The germinal centers, the lymphoid follicles will have germinal centers. All right. And then in these germinal centers, that's where you have your class switching and somatic hypermutation, because that's where the The B cells that are expanding and multiplying in those genes are the ones that are producing, undergoing class switching, somatic hypermutation, and also producing all the antibodies, all right, that will go out, release, and go into the bloodstream and fight the infection. All right. So class switching, somatic hypermutation done by B cells after they have been activated. All right. The whole essence of class switching, somatic hypermutation, is to produce antibodies that have a higher affinity for the antigen, will respond better to that antigen to stop the infection far better or faster. All right. So the other thing we talked about for T cell activation, we said, so I'm just going recapping now. So the T cell, for it to be activated, requires two signals, right? So the antigen present cell comes to the lymph node with the antigen, as you can see here, presents it to mount it on the MHC2 or MHC1, present it to the naive T cell via the T cell receptor, right? That's one signal. The second signal is between CD28 on the T cell and CD8 or 86 on the antigen present cell, or with B7. All right. So those two signals must happen for the T cell to be activated. All right. And once the T cell is activated, it now becomes uh, an activated T cell and begins to secrete all kinds of cytokines depending on the activating stimulus, all right, or the problem that's initiated its activation. Okay, so I think that's it. And so these co-stimulatory signals that we've talked about are very, very important, right? They're extremely important because anything that disrupts this interaction, this second, there are disease processes that can disrupt it, okay, and there are therapeutic processes that have been where drugs have been synthesized to disrupt that. All right. So I'll once we're done here, we're done with this page. We'll move to that. I'll take you to that page to show you diagrams showing these interactions and why it's clinically relevant. All right. So I don't know, any questions, comments, questions, things you need clarification on. All right.